the first session. The topic of the first session is DNA sequencing platform, which will be handled by our first speaker, Dr. Raj.
Thank you so much, guys, for joining this nice webinar organizing by the Genome Hub. So I'm here to just introduce something about the sequencing technology. I think most of the people who are working in the biology, they knows about. I'm just here to just polish some information which you have already. So let's start from the bioinformatics. Basically why we need the use of the bioinformatics. Basically, now the era is moving towards the big data, because already due to the immense use of the wet lab, there's a huge amount of data which needs to be analyzed. For a single person, for a group of people, like a small group of people, it's very hard to get the information or make the sensible information from the bioinformatics big data. So now the bioinformatics, basically it's going to help the biologists or other scientists to make the best knowledge or best result out of the wet lab informatics. Bioinformatics is the pure application of the computers in the field of to analyze a large quantity of complex biological data, and can be used to search biological database, compare the sequences, draw the molecular structure, and find the required information. So why we need the sequencing? Because as we know, all the information is stored in the DNA, and to understand all the physiological activities, if we know the sequence of the DNA from one end to another, that's going to help to better understand the all biological processes. As we know, the DNA is a blueprint of life, and the practical definition of the sequencing is to determine the number and order of the nucleotide that makes up a given molecules of the DNA. So the problem is to obtain the sequence of the nucleotide. As we know, the Rosalind Franklin discovered the structure of the DNA from the X-ray crystallography, and she got the Nobel Prize for this. And so DNA is a double helical structure. On the right hand, you can see, and the sugar and all the bases are attached with the double bond backbone. So we want to know what is the sequence, when the C comes, when T comes, and on which strand, positive or negative strand. So this, if we just want to sequence single strand or to find the information on this strand, so it likewise A, C, T, G from one end to on the last end. So basically from the double helical structure, we want to get the information in this form. So what is the inputs of the Human Genome Project? So it started in the 1990. In 2001, the draft genomes came up. Draft genome means basically the human genome means so it's called the draft genome. So day by day, the advancement of the sequencing technology. So the cost of one human genome is, when initiated, is the 3 billion. And 2000 it's become 300K. In 2008 become 100K, and 2010 it become 10,000, and 2011 it become 4,000, and 2012 to 2013 it's become 1,000. And recently, last week, I came up with the information. There are some technology and companies are coming to make possible the whole genome sequence in under $100. So if we see that from 90s to till now, from 3 billion to $100, it's a very a remarkable achievement in the scientific work. So after the sequencing of the human genome, we want to understand to get the more information, we want to understand about the different organism. Because if we see, there is more than 100 million species. If we want to see in the phylogenetically relationship, we want to understand how they evolve, what is the merging point. So how we can understand by doing the sequencing and comparing them, how close and how far from each other. if we see in the human population, we are around 7 billion. So we all share around 23 chromosomes, but we are different from each other in the name of the skin color, eye color, length, height, weight. So it is due to the single SNPs and other mutation. We can say the positive and negative mutation in our body. So that comes under the personal genomics. And as we know, we have around 10 to power 13 cells in human. So all these cells having some type of variation from each other, and that also leads to the cause some variation. If they are lethal, they cause the cancer and HIV. So we want to procure and we want to pre-predict these disorders. So that's why we want to take the help of the genome sequencing. So here are some queries that, for example, how many base pairs are in human genome? So in haploid genome, we have 3 billion base pair. So if we ask the next question, how much did it cost to sequence the first human genome? It's around $3 billion. How did it takes to sequence the first human genome? It's around 13 to 16 years. And as we know, the first human genome is completed in 2003. Whose genome was it is? Several papers, but actually mostly due from the buffalo. So as we know that the sequencing is an integral part of the research, and we need this sequencing to understand the genetic traits, how similar we are, how different we are. If see on the left side, there's a variety of dogs. So we want to understand what is the difference from these Poms to these Labrador or German Shepherd. To understand the differences and similarity, if we see the overview, just we need overview, we need to isolate the DNA and do the sequencing. And after that, if we see what are the changes in the base pair wise among all these 4 species, we can infer that why they are different from each other. And this will also give the answer of some animals or mammals, they are very closely related or different from each other. So role of the bioinformatics to understand the biological centered dogma. If we see, this is the DNA transcribed and make the RNA, and RNA then transfer the information in the translated form in the protein. And these are the proteins are what we can the slave or worker for every physiological activity. So the questions, what kind of scientific questions can we answer from the bioinformatics tool? So now an overview of sequencing data obtained in the research. So if this is just the general idea about how the sequencing things are working. So obtain the samples, like if you have any soil sample, water sample, blood, saliva, hair follicle, feathers, scales, which has the live system in it. So from that, we need to extract the DNA and do the sequencing by using the whichever technology you have, like Sanguar, Nanopore, PacBio. And after getting the data, these bioinformatician is going to help us to compare the DNA sequence, and they are going to answer our big question, how similar, how different from each other. So journal information about the sequencing, like if you are talking about the patient or the other samples, so it's just to collect the first sample, extract the DNA, sequence the DNA, and compare the sequencing. So the bioinformatician can understand in helping the large data set, because it's the cells have basically human body has a 10 to 13 power of the cells, and they have the different information. So it's a big data, it's a very big data. So manually it cannot be interpreted, or it takes hundreds of years. So that's why we need the help of computer and bioinformaticians, so they can make it easy for us. Now we can think about the sequencing technology evaluation. So sequencing technology till now divided in the three categories: the basic sequencing technology, advanced sequencing technology, and the next generation sequencing technology. So basic sequencing technology is a Sanger. It's a first generation sequencing or chain termination method. Second is a Maxim Gilbert or chain, and sorry for the confusion here. And second is the shotgun. So this divided in a shallow shotgun and deep shotgun sequencing. And third, or next gen sequencing, is divided in solid, Illumina, Pyro, PacBio, or third generation. Latest is a nanopore and PacBio. So by comparing these three, we can see that how we can use their their advantageous procedure. So the basic technology, the price of $1,000, is for a 1 million read, and advanced DNA sequencing technology is the price of $100 for 1 million reads. And the next gen sequencing, if we talk about the Illumina, it's around $10 for a 1 million read. But along all these three, the highest accuracy you will get from the basic technology is 99.99%. It's a very accurate, but it's a very slow. And the advanced technology, it's accurate, but it's faster than the basic technology. Next gen sequencer technology, it's less accurate, but it's also less time taking. So capabilities, if we talk about the capabilities of these sequencing technologies, Sanger can read the sequence from 600 to 1000 base pair. 445, this machine can read around 300 to 500. Illumina can read around 100 base pair. Iron Torrent can read around 200 base pair. And that what we can say, the PacBio and Nanopore, they have the ability to around 2000 base pair. But there are some other genomes, they have a more length of the DNA, like 3000 base pair, 5 million base pair, like a human cells has the 3 million base pair. So that's why we require the advanced technologies. So now let's talk about the first generation sequencing technology. So let's take example of the Sanger sequencing. So it's evolved in around 80s. So Sanger sequencing is known as a chain termination method. It's also a method for determining the nucleotide sequencing. The method was developed by the Nobel laureate Frederick Sanger. So it has the three basic steps. First step is DNA sequence for chain termination, PCR. Second is the size separation by the gel electrophoresis, and third is the gel analysis by the determination of the DNA sequence. So the crucial step is every step is crucial, but the most important is this first step. So the DNA sequence of interest is used to template for the specific type of PCR called PCR chain termination. So this is our query sequence, and this is our DNA template. So important things is here is the DDNTP, so dideoxyribonucleotide. So after adding these DNTPs in the appendrops or vials, we run the PCR, and DNA polymerase adds the DNTPs to the growing DNA strands by catalyzing the formation of the phosphodiester bond between the 3' OH group of the last nucleotide and 5' phosphate of the next. So in this process, these chains are moving along and stopped on like A stopped on the T, and opposite base pair of the T. So in chain termination PCR, the user mixes a low ratio of chain termination DNTP in with the normal DNTPs in the PCR reaction. So DDNTP's lack of the 3 OH group required the phosphodiester bond formation. Therefore, so if we see when the DNA polymerase incorporates a DDNTP in the random extension cases, the result of chain termination PCRs of the millions or the billions of polynucleotide copies of the DNA of the interest and terminate at the random length of the 5' DNTPs. So after the termination, we need to run the gel. So this gel is look like basically like this type. And after running the gel, we need to manually, on that time, these scientists are manually interpreting, okay, this is the C, and after that, this is the A. After that, if you see, this is the G, this is the C. So they are making from 3 to 5' end of the direction of the one strand. So the second is the Illumina sequencing. This is the advanced, we can say, and next gen sequencing. So we can see this is the My65 next gen sequencing Illumina and 500. So if we see how it advanced it and how it is helping in the advancement in the science. So one technician in around 30 hours in around $5,000 to $4,000, it can sequence this amount of the nucleotide. So this is the 40x of the human genome. So in which basically after the preparation of the libraries, this is the chip we need to load our samples on the one end in the flow cell, and it's around take 30 to 40 hours and depend upon the sample for the sequencing. So these are the basic steps for the Illumina sequencing. First is the library generation. So in which basically DNA fragmentation is going to be done, and on the end the adapter is added. See that this is your DNA. By ultrasonification, these DNA is sheared in the pieces and this adapter is added. After that, the cluster, after adapter generation, the cluster is formed. So this is the plate which is having the opposite sequence of the cluster. So the adapter is bind to on these plates small sequences, and after that adapter binding, there is a nearby adjacent molecule which is having the complementary sequence to the adapter. It's make a PCR bridge, and then so after the first round of amplification denaturation step, so if we see, this is make a loop and attach to this adapter, and so after that this is the one set of the PCR. So we can say is like a double strand of the DNA we are assuming, and after that this is the denaturation and amplification. So we can see this is the parental and this is the daughter strand, and after that these are the multiple cluster are formed, and by using the base amplification cycles, they are easily increased in the number. So after, and then the machine is going to use these fluorophore based sequence, and this is going, every fluorophore has specific for like every different nucleotide like A, T, G, C, and it's have the every different color. So machine is going to identify all these color, and then they are forming a DNA sequence. So third generation sequencing technology is an nanopore sequencing technology. So why this is a very important, and this is making the science on another like a higher level. This is a very portable sequencer you can have on your palm. You can use your laptop to analyze your data. You can take this sequencer to anywhere in your field, in your farm, in your lab, in your room. If you are traveling in your car, in your bus,